So this is the last video of Unit 2, Cell Structure and Function. And in this video, video 5, we're going over mechanisms of transport and cell compartmentalization, which is Units 2.9, 2.10, and 2.11. We'll also talk a bit about the origin of cells. So let's start with a review. Look at the pictures here and see if you can figure out which mechanism of transport it's representing. So name the mechanism of transport, state if it's active or passive, also state if it requires protein channels and/or ATP. So that first diagram is showing simple diffusion. So this is diffusion of nonpolar small molecules that can move directly through the membrane. So for example, oxygen and carbon dioxide are shown here. This does not require a protein channel, and because it's diffusion, it's going from a high concentration to a low concentration. It's going down its concentration gradient. It does not require ATP and is therefore passive. The next one shows the osmosis of water, or the diffusion of water, which is called osmosis. This does require a protein channel because water is polar. So polar molecules that are unable to get through the membrane on their own require a protein channel. But because it's still diffusion, it's going from a high concentration to a low concentration, this does not require ATP and is a form of passive transport. The other two pictures shown are active transport that do require ATP. So one example is a pump. So shown here is a sodium-potassium ion channel pump. And this is a form of a protein channel that moves sodium and potassium ions against their concentration gradient, and so requires energy in the form of ATP and is active transport. The next diagram represents bulk transport, so both endo and exocytosis of materials moving into and out of the cell. These do not require protein channels because it's movement through vesicles, but it does require ATP, and so is a form of active transport. So at the most basic understanding, a cell is a special place where the environment can be different on the inside of the cell, inside of the membrane, compared to the outside of the cell. You can actually have layers of compartmentalization within a cell. So take a look at these organelles and see if you can figure out what organelle they are, and also what do they have in common in terms of structure, and how does their commonality in structure relate to a commonality in function. So these organelles are the mitochondrion on the left and chloroplast on the right. Now, organelles by themselves are specialized areas within a cell, but these actually are compartmentalized even within the organelle. So within the mitochondria and within the chloroplast are small volume areas where you can get unique environments. So for both of these cases, there are small volumes that can build up a high concentration of protons, and then that allows the production of ATP. So there are areas that have a high surface area to volume ratio within the organelle, which is within the cell. We'll get more into this in the energy unit, but for right now I just want you to understand that compartmentalization has sort of multiple layers. So there are special areas within special organelles within cells which are special to begin with. So where did these first special cells come from? How is it that we first had a substance that could be different on one side of a membrane than the other? There's a great video entitled Where Did Life Come From that I highly recommend, and it talks about the origin of life and the origin of cells. So cells at their layer, their membrane, has a polar and a nonpolar area. By themselves, without any input of energy or anything being alive, those molecules can form essentially a bubble. So just from the molecules themselves, you can get a lipid bubble. Now you combine that with some production of protons, and you essentially can have a specialized contained area, which is basically what a cell is. So what we do in our cells is we create that proton gradient using the energy from food. Plants use the energy from sun. But the very first cells didn't have access to either of those. What they did have access to were deep sea vents that produced protons. And so these self-forming bubbles could form around these deep sea vents and fill with protons. And that was essentially the very first cell. There certainly are major differences between the cells that we have in our bodies and those very first rudimentary cells. So there were a lot of important steps along the evolutionary history of cells, but one of those was endosymbiosis. So the idea of endosymbiosis is that very early cells, very early prokaryotes, actually one of them engulfed another cell, and that smaller cell was then essentially living inside this larger cell, and that benefited both sort of cells, because then you could get specialization within the cell, and so that was essentially the very first organelle. So this idea of endosymbiosis, where you have the history of chloroplasts and mitochondria are bacterial cells, this is called the endosymbiotic theory. Now I want to be clear about the word theory because it means something different in science and everyday life. So what does the word theory mean in science, and how does that differ from how it's used in everyday life? So the term theory in science is an explanation of some aspect of the natural world that has been substantiated through repeated experiments or testing. In other words, it is well established and it's based on good evidence. And that is true of the endosymbiotic theory. Some of the pieces of evidence supporting the endosymbiotic theory include looking at membranes, looking at DNA, and looking at reproduction. So in mitochondria and in chloroplast, those organelles have their own membranes, just like prokaryotic cells do. And this is evidence that they once were, evolutionarily, their own cells. Another piece of evidence comes from looking at DNA. Mitochondria and chloroplasts as well have their own DNA. So it has a circular DNA genome, much like what you see in bacteria. And this is then passed from mitochondria to mitochondria. So you actually, in your mitochondria, have special DNA that's separate from the DNA in your nucleus, and that came from the mitochondria that your mother gave you. And we'll get more into mtDNA in a future unit, but this is another piece of evidence supporting the endosymbiotic theory. Finally, another piece of evidence supporting this theory is reproduction, that mitochondria multiply in the same way as bacteria do, and chloroplasts as well. So each mitochondria, in order to make a new mitochondria, it has to come from an old mitochondria. Just like in cell theory, that all new cells come from old cells. So mitochondria cannot be produced by a cell. They have to come from other mitochondria. And so that's another piece of evidence supporting the endosymbiotic theory. So that's it for this unit. So we studied cell structure and function, and I hope you've enjoyed learning a little bit more about cells.